Let me introduce Professor Francesca. She is an assistant professor in the Department of Biomedical Engineering at Eindhoven University of Technology. And her team works at the intersection of machine learning, chemistry, biology, and her group focuses on designing innovative AI approaches for molecular discovery. So today she will be talking about charting the dark chemical universe with deep learning.
Thank you very much for the nice introduction, and I'm really sorry that I couldn't be there in person. Hopefully, the next time I will manage to be there. So, as I said, today I will talk about how we can use deep learning to chart this dark chemical universe, especially when we have very little data available for training. If my PowerPoint slides collaborate, yes. So I think you are already familiar with the idea of what the chemical universe is, or what we call a chemical universe. And this is that virtual space where all of the possible drug-like molecules that we could in theory make in a lab and test are contained. And this chemical universe is estimated to be extremely large. So for example, to contain up to 10 to the power of 60 molecules. And in contrast, as humans, we have only discovered about 10 to the power of 4 small molecule drugs. So this universe is extremely vast. It contains way more molecules than there are stars in the Milky Way or cells in a human body. And most of it is completely dark to us. And this is why machine learning has become so attractive to really try and navigate this chemical universe in search for interesting molecules such as drug candidates. But actually, compared to other fields where machine learning and deep learning really have led to incredible breakthroughs, there is an additional challenge when it comes to drug discovery. And it's not only that this chemical universe is really, really vast, so that we are dealing with really, really small data regimes and to try and navigate something that is much, much bigger. But also we have other troubles that relate to it being a very low data endeavor. So for example, if you think about fields like playing very complex games, for example, chess or Go, where deep learning has led to incredible breakthroughs, when we're dealing with discovering drugs, so for example, when trying to design molecules that bind to a specific target, we do not know the rules of the game we're playing. So we're really trying to learn something that we actually as humans don't know, so we cannot infuse it in our algorithms. And finally, we also have quite a problem when it comes to translating complex chemical information for a computer to learn from. So you all know that, for example, a ligand binding to a protein is a very complex and very dynamical problem that is also very 3D. But in many cases, we really fail or struggle to capture this complexity when it comes to translating it into, for example, molecular representations. So I would say this is much more challenging than playing chess, and also much more challenging than training algorithms to learn how to play chess better than humans. And probably this is also what makes this field incredibly exciting as well. So there are many flavors of how we can apply machine and deep learning approaches to try and find interesting molecules. And I would say that in many cases, or in the simple case, we can think of it as just a mapping problem, right? Where we have a set of molecular structures and we have some properties we're interested in, and we basically can train machine learning models to try and perform some sort of mapping. And in the traditional way that is quite well established and has been there for decades, we can apply machine learning, or what was once called quantitative structure-activity relationship, in, let's say, the forward way. So we basically build predictive models where we can link structures to properties such as bioactivity, and we can use these models to screen large libraries of molecules that we could in theory purchase and make in a lab and test. And I would say that in this domain, more complex algorithms, more sophisticated algorithms like deep learning algorithms really struggle to outperform very well-established cheminformatics tools and traditional machine learning approaches. But there's actually another direction, which is the, let's say, reverse direction, which is called de novo design, that actually deals with, instead of using models to screen large libraries, it deals with designing molecules on demand from scratch that hopefully satisfy certain properties. And this, I think, is one of the fields where we really see the progress that deep learning has brought compared to more traditional approaches that have been there for a few decades. And even here, you can think about many different algorithms and many different approaches to design molecules from scratch, for example, with generative deep learning. Let's say that already a few years ago now, it starts to be old stuff in deep learning terms. I started to work on a specific branch of generative deep learning approaches together with my colleagues at ETH first and now with my team here in Eindhoven. And this subfield of generative deep learning for chemistry is often referred to as chemical language modeling. So for some of you, it might be strange to find this pair between chemistry and language modeling, but if you look at this molecule, you could see that there is such a thing as a chemical language. So if, for example, I asked you to connect these atoms randomly to each other, you would probably know that we will end up with a molecule that does not make much chemical sense. So there is a syntax or a grammar to what it means to be a molecule. And at the same time, when we want to design molecules from scratch, we're not only interested in making molecules that make sense from a chemical standpoint, that they are reasonable, but what we mostly care about is to get the right semantics, so to get to the right properties, for example, in our case, to bioactivity. So we can think about this analogy and think about this chemical language and in turn use, for example, string notations that represent molecular structures and use sequence-based approaches to model these sequences, for example, these smile strings that you see here, and use these representations to generate new molecules in the form of smiles. So this is not a new concept anymore, and there are many ways of using these string representations that capture 2D information to generate new molecules. And historically, one of the most, let's say, widely adopted and also experimentally validated approaches is constituted by the so-called neural networks with long short-term memory, so the so-called LSTMs. And I guess most of you know how these networks work, but in case you don't know, they're quite simple. So the idea is that you can have a set of smile strings. Often you attach a go and an end of the sequence token or character, and you just basically can train these networks to predict at each step of the sequence which character it should follow. So if you have enough data and you train these networks for a sufficiently large number of epochs, what you hope to achieve is that the model will have learned the underlying probability distribution of a certain token to follow a certain portion of your smile string. So then, once the model has been trained, it can become a generator. So you can basically start from the go token, let the model generate the next token, which becomes the input at the next step, and so on and so forth until the end of the sequence token has been reached. And this idea was published for the first time around 2018 by Marvin Ziegler and co-authors. And it showed that actually we can use these models to really generate new smiles that seem to make sense from a chemical standpoint. So back when I was a postdoc at ETH, we thought, okay, can we actually bring these models to the wet lab and design for the first time molecules that are bioactive on an intended target? But then, of course, as almost always in drug discovery, what we were facing was a lack of a sufficiently large amount of data, for example, of bioactivity data on a given target to train a model from scratch. So there are many flavors also here of what you could, in theory, use to overcome this lack of data. And what we relied on here is the so-called transfer learning. And the idea is very simple. So if you do not have enough data to train a model from scratch, a focused model from scratch, you can resort to some data that somehow relate to your problem to pre-train a model, and then you take this data away, bring in your data, and you can fine-tune the model to hopefully make it more focused on the task you want to achieve. So back then, we decided that since we wanted to design bioactive molecules, we could pre-train with the smiles of bioactive molecules from Campbell, regardless of the targets they were annotated for. And we already were seeing that after this pre-training step, actually, if we just asked the model to generate smile strings, it would get to more than 90% chemically valid smile strings. So just with this step, these models, very simple models, can learn the grammar of what it means to be a valid molecule. But then, of course, we were interested in achieving bioactivity. So we thought, okay, let's take out this data and bring in a very small set, actually, of modulators of nuclear receptors, which we used, or the smiles of which we used to fine-tune our model. So then, once the model was fine-tuned, we just produced 1,000 smile strings, which we ranked with our usual procedures, and then gave them to the chemist and said, okay, could you please start from the top, make the top 5, and just test them in the lab for bioactivity. And actually, I have to say that back then I was not expecting to get results that were so good, because basically these are the bioactivities of the 5 molecules that were tested in the lab. And what you can see is that 4 out of the 5 molecules actually showed quite good biological activity on the intended target. And this was impressive back then, because if you think about it, the model just saw a bunch of smile strings, but it still managed to capture very complex information that you need for a molecule to bind to a macromolecular target, like in this case, nuclear receptors. And also the fine-tuning set was really, really small if you think about it, because it was only 25 small molecules. And recently, we collaborated again with the chemist that back then was a colleague of mine and synthesized those molecules, because in his lab, they discovered the first agonist of NUR1, a nuclear receptor. And basically, he was wondering whether we could use just a single molecule to fine-tune these models and see whether we can actually generate more agonists. So we followed the usual procedure, and actually what we discovered is that no, we cannot, or at least this very simple pipeline did not work, in the sense that it was really difficult to generate molecules that were not very trivial modifications of these single agonists, or molecules that were not making much chemical sense from, let's say, a medicinal chemistry standpoint. But then what they did was to fine-tune again with some weak agonists that they had laying around in the lab. And actually, they were able to design some more NUR agonists, so 2 more very potent NUR agonists, NUR1 agonists, only with this very, let's say, relatively easy way of thinking about how we use this fine-tuning. And this was just to show that actually these chemical language modeling approaches are very applicable and are also quite flexible to be applied in the lab to design bioactive molecules, even when we bring down the number of fine-tuning data that we have available. And actually, since I was at ETH as a postdoc, we have applied these models to a bunch of tasks, usually in quite a low fine-tuning data regime. And also, you can think that these models can be applied to anything that can be represented as a string. So, for example, we also applied it to the design of anti-cancer peptides. So a few years ago, then I moved to the Netherlands to lead my team, and we're also quite interested in bringing these models to the wet lab, but we're also interested in trying to stretch the boundaries of what these models can achieve when applied to the discovery of interesting molecules and when applied to exploring this vast and dark chemical universe. So one of the things we did was to actually go back to the whiteboard and start thinking about what we're actually trying to tackle as a problem with these models. And so, for example, I told you that we can represent these molecules in the form of strings. I didn't tell you exactly how it works, but basically the idea of these smile strings is that you start from a molecule in the form of graphs, so 2D information, and then you basically start traversing the graph and annotating what atoms you find on your way, on your traversal. But actually, you might probably see it already from this figure. Very often what happens is that because we linearize a complex structure into a single string, what happens is that very often atoms that are very close together in the actual molecule end up very far in the sequence. And also often when we deal with these LSTMs, with these networks, what happens is that the more you learn from the sequence, the more you start forgetting what you saw before. So the first parts of these SMILE sequences. And we can think that whenever we deal with bioactivity, you might have properties that depend on parts of the molecules that are maybe very far away from each other in the SMILE string. So we thought, okay, are LSTMs the best way to go? Or should we maybe rely on something like transformers, which instead of looking at one character at a time and generating one character at a time, they actually see a big portion of the SMILE string to decide what to generate next. Maybe they will capture bioactivity or complex properties better because they have access to the full string. So Reza, one of the first PhD students in my team, started to really do a very systematic benchmarking study where he was comparing transformers and these LSTMs on a wide array of properties, for example, in terms of learning bioactivity. And when we look at the ability to learn bioactivity, maybe this graph is a bit complex, but you find the details in the paper, we see that seeing the entire sequence at once actually leads to a model that learns bioactivity better. And we can imagine that this is because the model has access to different parts of this mice sequence and also different parts of the molecule that might be important. However, when it comes to designing molecules that are structurally novel, which is one of the goals because we want to go out and explore this uncharted chemical universe, generating one element at a time or one atom at a time actually works much better than using a transformer to generate a full sequence at once. And then also, depending on what you're looking at, whether it's the training time or generation time, each of these two approaches have advantages and limitations. So for example, transformers don't scale very well when you go to quite long smile strings. So then Brizard started to dig into the literature and asked himself whether we can actually find something that makes the best of both worlds and doesn't force us to compromise on either aspect of either generating structurally novel molecules, but also capturing bioactivity. And he found this family that a couple of years ago was basically very new, which is the family of the so-called state space models, and in particular a type of architecture called S4, which is structure state space sequence model. And the interesting element about this architecture is that during training, it can be formulated as a global convolution, so a model that has access to the entire sequence at once. But with the same set of learnable parameters, when you use this model to generate, the authors demonstrate that it's actually like generating one character at a time in your smile string. So a bit of a combination between both types of approaches. And when we add this model to Brizard's benchmark, what you can see is that actually when it comes to bioactivity, S4 outperforms both the other models, so both holistic and sequential models. And when it comes to generating diverse smile strings, it also performs similarly, although not better than LSTM. So really, by combining these two aspects, we can make the best of both worlds. And you can also see it when you look at training time and generation time. So of course, what is also important is to see whether we can apply these models in practice in a prospective way. So although we didn't bring these models to the wet lab, we applied it prospectively to generate kinase inhibitors with the same approach I showed you before, so via pre-training and fine-tuning. And what we could see is that in our top 10 designs, we actually found molecules that were predicted to bind to the intended target in a similar way or even with a better affinity than the molecules we had in the fine-tuning set. So this shows that there's some benefit to using these models or bringing these models into chemistry. And also what is quite interesting, I think, is that I showed you that, for example, transformers don't scale very well to very long sequences. And in general, these state space models were developed to deal with very long pieces of text, of natural language. And we also see that they work quite well when we apply them to very long smile sequences. So for example, in this case, we apply them, we use them to generate natural products. And you might know that natural products are quite interesting as a source of inspiration for medicinal chemists. And what we see is that when we look at the capacity of these models to match the distribution of some of these complex molecular properties, S4 outperforms all of the other models on all of the metrics that we tested. So it shows that really, even when you have very long sequences, it's able to capture elements that might be quite far apart in these smile sequences. So this is one of the approaches that recently came out of the group, and we are now thinking about bringing it to the wet lab for a final validation to really see whether these models are as good as they seem in capturing both structural novelty and complex molecular properties like bioactivity. But also what we're really interested in is to try and see how we can push these models to work, for example, in cases where we have very little data or even no data at all. So one of the concepts we have been playing with for a while now is the so-called task arithmetic. And the idea of task arithmetic is really to try and edit the inner, let's say, weights of a model. So how does it work? You can think of a model as a point in this multidimensional weight space, right, where every model has its own weights. And for example, when we start from a pre-trained model and then we fine-tune it on a given task, what we obtain is what we call the task vector. So the task vector represents how my model moves in this weight space when it's fine-tuned. So it's something that represents the learning of the task itself. So a relatively recent paper applied this so-called task arithmetic and started from a pre-trained model that was a language model. And this language model was fine-tuned on toxic text, so like toxic language. And they could show that if you take this task vector and flip it, so you basically move in the weight space in the other direction, the effect that it has is that you remove toxic language from your model. So basically you negate one property, so you bring the model in the exact opposite direction. And this idea was very attractive to us because we thought, okay, what if we could use it to remove properties we do not want from molecules, or for example, to learn from negative data to generate positive data. So this is the idea, what we now call molecular task arithmetic. And the idea is, could we use this strategy to really perform zero-shot design? So to start with no positive molecules and generate positive molecules. So what result did was to analyze a bunch of molecular properties. And for example, we started from relatively easy descriptors that can be easily computed. So this makes our life a bit better when it comes to analyzing what these approaches can do. And so, for example, we analyzed log P, the fraction of sp3 carbon atoms, and so on and so forth. And the idea was, what if we used molecules with a high log P value for fine-tuning, and then we flipped, would we obtain molecules with a low log P value? And what if we do the other way? What if we apply the models the other way around? Would we still be able to flip and obtain molecules that make sense from a chemical standpoint and molecules that have the desired property, despite the model was never exposed to the data with those properties? And how Riza calls it, could we look the other way to generate positive molecules from negative data? And actually what we saw was quite interesting. So you see here a bunch of properties, and the arrow indicates whether we want to maximize or minimize this property. And in gray, you see the properties of the pre-training set. And here you see the performance in terms of success rate. So how many molecules that are generated that pass a certain threshold. So you see that, of course, and here, this is the fine-tuning, so with positive molecules, and this is the molecular task arithmetic, so trained with negative molecules, so molecules having the opposite property we're searching for, that then get flipped and then are used to generate molecules. And what you see is that, as expected probably, when you fine-tune with positive data, you actually do better in terms of overall percentage of success. But still, molecular task arithmetic on some tasks seems to work better than just trying to pick from a pre-training library. But what was more interesting to observe is that when we then cluster our designs in terms of diversity, so we look at how many different hypotheses the model gave us in terms of molecules that still pass our success threshold, we see that molecular task arithmetic actually gives us more options to choose from. So more molecules that pass our criteria or criterion, that are still more diverse, that are diverse from each other, more diverse from each other. So basically, more ideas for us to pick from. And then, of course, one of the first criticism of this is that we're dealing with very or relatively easy properties to model, which we can consider linear, right? So you can, if you add certain substructures, your log P will increase. If you remove them, log P will decrease. So, but what happens when we actually try and think about bioactivity? What does it even mean, for example, to train with negative data, so inactive molecules, and try to generate positive molecules? Is it even possible? So it's almost a philosophical discussion here. So what Reza did was to then resort to docking. And the idea here is, can we use molecules that dock very badly on a given target and use them to train a model and then flip it? Would we generate then molecules that dock better or well on our target? And although this is not like going to the lab, we know that this is still quite a complex property to learn, because still the docking scores depend on shape and size, molecular flexibility, the presence of some key interactions, and so on and so forth. So it is still a good proxy to see what happens when we flip these models in the weight space. So what we see is that already when we do molecular task arithmetic, we do better than, for example, using only a pre-trained model to generate. So it's better than just saying, okay, I know nothing, and I just enumerate a library as large as I can, and then I try to dock it. If you have some negative data or produce some negative data, then you can flip your model and you will do actually better. But of course, when you do have positive data, so you do have molecules that dock well or are active, you actually can perform quite well or better than molecular task arithmetic in terms of your success rate. So in terms of how many molecules were actually dock well in terms of a given threshold. What was interesting to observe for us was that if you combine molecular task arithmetic and fine-tuning, so you first do this fine-tuning with negative data, and then you flip, and then you fine-tune with positive data, you still end up with a more diverse set of candidates that are successful. So a broader exploration of the chemical space while still being able to find molecules that indeed pass your success criterion and dock well. So for us, these are still unpublished data, but for us, these results are very encouraging and very interesting to observe that one could still generate valid molecules that also match certain criteria, even when the molecules, when the models have not been trained with positive data at all. So I think there's a lot of potential to explore when it comes to these model editing techniques to explore the chemical space. And then I would like to show one last study about coming out from a group. And this is really about trying to explore the chemical space and know whether we can trust what the model produces in those regions that are relatively unexplored. And this is a term, the term is like going beyond the chemical Kármán line, which was coined by Derek, a PhD student in my team. And for those of you who don't know what a Kármán line is, this is considered to be the surface that separates what we consider the atmosphere from what we consider the outer space. And basically, that's really what we want to do when we try to use deep learning to explore the dark chemical universe. We really want to go beyond this chemical Kármán line to find molecules that we can trust and bring to the lab. So normally in the cheminformatics community, we assume, well, in every application field of machine learning, what we know is that models tend to not work very well when applied to molecules that are outside of the distribution of the training set. So very often when we use these models, what we do is that we say, okay, whatever is too far or too dissimilar from the training data that we've used to train our model, we cannot trust. So those molecules or those predictions we just consider to be out of the applicability domain. And then we also have approaches that are actually more model-centric rather than data-centric. And these approaches, for example, uncertainty, tell you, okay, this is how much you can trust my model prediction. But also in the case of uncertainty, what we know is that very often these estimations might be overly confident, even when it comes to predictions that cannot, in theory, be trusted. So what Derek did in this project was really to try and think about a novel way to capture how much we can trust these predictions, to try and find the sweet spot between predictions I can trust, but also on molecules that are sufficiently diverse from my training set to be interesting for a drug discovery community or a medicinal chemistry project. So in this case, what we did was to go back to still use generative deep learning, but going back to virtual screening. So how we can use it to screen large libraries of molecules that we could, in theory, purchase and make and test in the lab. So what it did was to actually rely on a rather simple encoding-decoding architecture. And I guess all of you are familiar with this. So we start from a molecule represented as a smile string. We have an encoder that converts this molecule into a vector, a latent vector, and then a decoder that basically reconstructs this molecule into its original version. And then to this type of networks, we can also attach a prediction head that would predict a property, in our case, bioactivity, of a given input molecule for you. So these approaches have been used mostly for de novo design because they're really nice, since you can generate molecules and also predict their activity. But in this case, we thought, okay, could we use these approaches for virtual screening instead, so for predictive modeling, and use the information that our model gives us in terms of how well it can reconstruct an input molecule as a proxy to understand how much a model understands the chemistry of that given molecule. And the hypothesis here was that if a model cannot reconstruct the input molecule well, then we can also not really trust the bioactivity that the model predicts. And basically, this reconstruction loss is then recast into a metric that Derek calls unfamiliarity. So the higher the unfamiliarity, in theory, that was our hypothesis, the least we can trust, the less we can trust the prediction of our model. So, and of course, we first went into a big computational study where we analyze what this unfamiliarity really captures. And we do see that unfamiliarity correlates, wait, let me move my face out of the, okay. So that actually unfamiliarity correlates quite well with some metrics of accuracy, so how well the model actually performs. And it's not too different from a well-established metric like uncertainty. And what you can see is that the, let's say, well-established and traditional chemoinformatics approach where you say, okay, everything that is too dissimilar, I don't consider as reliable. These metrics do not, these approaches do not correlate too well with performance. But then we know that also this reconstruction captures also elements of the structure. And in fact, sorry, this doesn't work anymore. Yeah. And in fact, when we look at how much of chemical information this unfamiliarity captures, we see that it also correlates with aspects like scaffold similarity, presence of certain pharmacophores. So we thought that this is somewhere in between something that captures performance, but also something that captures structural information. So we thought, okay, why don't we use this? Because this is exactly what we want to do, right? We want to find molecules that are structurally novel, but at the same time reliably predicted. So could we use this unfamiliarity to guide the exploration, for example, of a large virtual library? So what we did was really to apply this model prospectively to screen a large library of 1.4 million molecules. And what you can see here, I'm not sure how clear it is, but basically you see the comparison of this library in terms of a few metrics, so our unfamiliarity, uncertainty, and the similarity to the training set. And you have library in green and two test sets obtained from our data, available data, in yellow and blue. And what was surprising to us was to see that if you look at similarity or uncertainty alone, this library seems to be more or less in line with the data that we had available. While when you look at unfamiliarity, which probably combine aspects of both the other metrics, you see that some molecules seems to be more further away from our distribution than when you just look at these the first two metrics. So we thought, okay, why don't we use unfamiliarity to try and really find those molecules that are diverse enough from our training set from a structural point of view, but also reliably predicted, so with a very low or as low as possible values of unfamiliarity. And that's what we did. And then we picked the top molecules and we brought them to the wet lab, or Derek, not me. I don't enter a lab, I'm very dangerous. And what he found was that we applied it to two protein kinases, and what he found was that indeed some of these molecules that were highly familiar, but also quite diverse from the training set molecules, were actually active in the low micromolar regime. So this really shows that with this new metric, we can really navigate the chemical universe and try to find that sweet spot where we can trust our predictions, but still find interesting chemistry. And I hope that this metric will be progressively adopted by the community to see how much we can really use it and whether it really works in multiple scenarios. So all in all, I think this is a very, very interesting time to be working in this domain. And I think that some of the directions I showed you really hold a very big potential. So I think there's an incredible value in trying to infuse more sophisticated or learn more sophisticated chemical information from the data we have available, and also trying to go towards learning from very small data sets or even cases where we do not have any positive example at all. And ultimately, what we hope to achieve is really to find better ways to identify molecules that are at the same time structurally novel and interesting, but also possess one or more properties that we are interested in. So there's much more research that is going on in the team that I couldn't present today, somewhere at the interface between learning only from ligands, learning from proteins, or from the interactions of both. So if you see anything that looks interesting for you, just feel free to write me a message, and I would be happy to chat about science. I'm very sorry to not be there today. And with this, I am done. Of course, I want to thank the organizers for inviting me, the team who's actually working really, really hard to produce the research I'm showing you today, our collaborators and the sponsors, and then of course you for the attention. Thank you.